Hello, dear students. I welcome you all on behalf of Sri Jendra Rangjung classes. Dear students, we are in a continuation of the chapter that is plant growth and development, and we are studying now on an important topic that is plant growth regulator. In this, we have already discussed about the two promoters, that is plant growth promoters, that is auxin and gibberellin in the previous lectures. Today in this lecture, we are going to discuss one new plant growth promoter under the regulators, that is cytokinin. Okay, so first we should note here that it is also a plant growth regulator. Okay, now we are going to discuss about this particular hormone. So cytokinins, as the name suggests, cytokinins means cytokinesis promoting hormone. We can say simply that they are the cytokinesis promoting or cytoplasm division promoting substances or hormones are the cytokinins. Now, chemically it is an adenine derivative, means the precursor for the cytokinin is the adenine derivative, that is N6 furfuryl aminopurine, and which is known as kinetin. Okay, so N6 furfuryl aminopurine is a nothing but a kinetin. Okay, now here F. Skug is the one of the important scientists in the discovery of this particular kinetin, or we can say cytokinins. And what F. Skug did, that we have to understand here. Then it is known that F. Skug and his co-worker observed that from the internodal segment of tobacco stem, if we take a tobacco stem and if we take an internode segment, the callus get proliferate from this internode segment, that is an undifferentiated mass of cell, if in addition to auxin the nutrients medium was supplemented with one of the following extract. Means along with auxin, if some new supplement extracts are added, then this tobacco callus is going to proliferate. And the particular extracts are vascular tissue, it can be yeast extract, it can be coconut milk, means we have to provide a nutrient mineral along with the auxin with the extract of the yeast, extract of the coconut milk, or vascular tissues, or we can say the DNA. If you provide this many things along with the auxin, then only the internode segment of tobacco get form a callus or get proliferated. So Skug was identified this, and they were analyzed to find out the growth promoting chemical. This all substances was identified to understand the growth promoting chemical. But the Miller was also joined the company of Skug, and later on the Skug and Miller identified and crystallized the cytokinesis promoting active substance that they termed kinetin. That they termed kinetin. So cytokinesis promoting substance identified and crystallized and was named as a kinetin. And kinetin is obviously nothing but a N6 furfuryl aminopurine. Now this is the scientist who worked over this, Cook and Miller. Now cytokinins obviously have a specific effect on the cytokinesis, means they promote a cytokinesis, a cytoplasm division, and they were discovered as kinetin, that is nothing but a modified compound of adenine, that is a purine, nitrogenous base, from the autoclaved herring sperm DNA. Means this first cytokinase was discovered from the degraded autoclaved herring sperm DNA. Means if we take a herring fish and if we take about sperm DNA, then from this the autoclaving of this done. Autoclaving is a particularly heating them at a high temperature and pressure in an autoclave machine, and from that the adenine was obtained, and from adenine this kinetin was obtained from that particular adenine of DNA. So this is cytokinesis promoting, or obviously this is derived from the sperm DNA. Now kinetin does not occur naturally. One more important point that you should remember, that kinetin is not going to found in the plant naturally, and search for natural substance like cytokinin, natural substance with the cytokinin like activities led to the isolation of zeatin from the corn kernels or a coconut milk. So first natural cytokinin, if we talk, that is obtained from the unripe maize grains, that is corn kernels and coconut milk. Okay, we can corn kernels or we can write unripe maize grain and coconut milk from this by Latham et al. In 1964, and it is known as zeatin. Obviously, if we talk about the zeatin is nothing but 6 hydroxy 3 methyl trans butyl aminopurine, and this is a natural cytokinin. Whereas the kinetin is obviously not occur naturally, it is artificially obtained from the DNA of herring sperm DNA. Now this kinetin also occur in coconut milk, obviously. So natural cytokinin are synthesized in the region where rapid cell division occur. So the synthesis of this natural cytokinin or zeatin occur in the regions like rutabasis, developing shoot buds, young fruits, etc. And it is known that these are the obviously division promoting substance. So fruit, we can say young shoot buds, developing apices, rutabasis, young fruits, that all should have this particular thing, that is zeatin. Now it is known that up to 18 types of cytokinin have been discovered. 18 types of cytokinin have been discovered. Now the cytokinins are basic in nature. Cytokinin are basic in nature. If we talk about oxygen and gibberellin, which we have discussed, they are acidic. So either they promote a cytokinesis, they mainly promote cytokinin cell division either alone or in a conjugation with the oxygen, that we have discussed, and how the discovery occurs, that also we have found. Now, in the cell dividing regions, these roots seem to be major source of cytokinin. Major source of cytokinin synthesis. And from root, the cytokinin pass upward through the xylem. So from root, cytokinin pass through xylem. And some cytokinin synthesis also takes place in other areas where cell division occurs. There is the endosperm region of seeds, growing embryos and developing seeds, young fruit, developing shoot bud, that all thing. And coconut milk is also a rich source of cytokinin. Coconut milk is also a rich source of cytokinin. So that is what about the discovery we can say. Now we talk about the functions, and if you talk about the functions of cytokinin, then the first function is it helps to produce a new leaves. It helps to produce a new leaves. Then the number of new chloroplast in the leaves, lateral shoot growth, and adventitious shoot formation. This all things happen in the particularly we can say during the differentiation, this cytokinin induce formation of this all thing. Okay, so that is the first thing that happens. So new leaves, chloroplast in leaves, lateral shoot, and growth of cytokinin also help to overcome the apical dominance, which is the second important function of this cytokinin. That is, it promote apical dominance. Means if we cytokinin concentration is reached in particular plant or in the dividing cells, in the presence of apical bud, also the lateral bud will grow. So what they promote? They promote nutrient mobilization. The second thing is they promote a nutrient mobilization which help in the delay of leaf senescence. So proper nutrient availability can be carried out by this one. Okay. Then the point we have discussed, that is apical dominance just now. So what it is saying, the presence of cytokinin area causes a preferential movement of nutrients towards us when applied to a lateral bud. They help in their growth. Okay. So if cytokine is applied to little bud, the little buds start growing. Okay. So in a apical dominance, it is known that cytokinin counteract apical dominance. Means it remove apical dominance. Means when cytokinin is applied to lateral bud, it help their growth or allow their growth despite the presence of despite the presence of apical bud. So we can say cytokinin act antagonistically to auxin in a reference to the apical dominance. Okay. Then the fourth thing, it promote cytokinin promote cytokinesis. Though it promotes cytokinesis because in absence of cytokinesis, chromosome doubling can occur. It is also known that cytokinin bring about division in presence of auxin. Cytokinin bring about division even in permanent cells. Even in permanent cells. And obviously, cell division in callus, that is undifferentiated mass of undifferentiated unorganized mass of cell require both hormone. So if we talk about a tissue culture, if auxin is more in concentration than the cytokinin in tissue culture, the root initiation take place. Whereas if cytokinin is more than auxin, then shoot initiation take place. Whereas when the concentration of auxin and cytokinin is equal, callus formation occur. Okay, so such a way they help in a particularly differentiation, or we can say the tissue culture thing. Next function, if we talk, then it is cytokinin also causes cell elongation. Also cause cell elongation. And next, also bring about lignification, lignification and differentiation of interfascicular cambium. Then eighth thing is senescence. Eighth point is senescence. And in this, it is known that cytokinin delay the senescence of leaves and other organ by mobilization of nutrients. So mobilization of nutrients can occur due to this the senescence, that is the end, the time period before the death can be extended. Then like gibberellin, cytokinin overcomes cell dormancy, means it promote the seed germination. Okay, so it overcomes seed dormancy of various type, including of various type, including red light requirement, of lettuce and tobacco seeds. Then tenth is, it is known that cytokinin increase resistance to high or low temperature and disease condition. Then Cytokinin also help in phloem transport, means it promote the transport through the phloem. Then, if we talk about more effect of cytokinin, then it is known that it induce accumulation of salt, accumulation of salt inside the cells. And 13th is, it is important point to note that cytokinin can replace photoperiodic requirement of flowering in certain cases. In certain cases. And 14 is sex expression, where it is also known that cytokinin promote femaleness in flowers, means it promote the development of female flowers. Then it also induce parthenocarpy, that is the development of seedless fruit through the cytokinin treatment. Then it is essential, as we have discussed the role of oxygen and serotonin, it is essential for tissue culture to promote the root and shoot initiation. Then cytokinin, it is known that it can, cytokinin can prolong the shelf life of cut shoots and flowers. It can prolong the shelf life of cut shoots and flowers by employing the hormone, by employing the hormone, that is cytokinin. Now it increases the shelf life, means application of cytokinin marketed vegetables to marketed vegetables can keep them fresh for several days. So this application of cytokinin can make a marketed vegetables fresh for the several days, we can say. Then we have discussed that it delays the result. So these all things are the important uses and applications and functions of the cytokinin. Now we are going to last talk about the bioassay of cytokinin. If we talk about a bioassay of cytokinin, then first bioassay, that is tobacco with culture test. And it is known that out of the two tobacco with cultures, one is supplied with the cytokinin while the other is not. So increase in fresh rate of the tissue over the control is a measure of stimulation of cell division and cytokine activity. And through this tobacco pith culture test, by providing one with the cytokinin and without the cytokinin, the test measure a cytokinin concentration between 0.001 to 10 milligram per liter, and it takes obviously 3 to 5 weeks. So this detailed information is not need to remember, but you should remember this tobacco pith culture test are particularly we can say the bioassay of the cytokinin. Second, so first bioassay is a tobacco pith culture test. Second one is excised reddish cotyledon expansion test. So in this, the test was developed by Latham, and in that excised reddish cotyledon were measured and placed in test solution as well as ordinary solution. The enlargement of cotyledon observed in the particular test where cytokinin was present. So through this, we can carry out an enlargement of cotyledon, which is an indication of the cytokinin activity. And third is the retardation of leaf senescence. So these are the some of the important we can say the bioassay of cytokinin. So I hope you understood well of the cytokinin, and that is the third important plant growth promoter which we have discussed. Okay, so I hope you understood well. Thank you everyone.